Hello and welcome to my latest blog. Today we're going to talk about sequencing and how we can harness the power of genomes to control infectious diseases. Sequencing is a technology that's been around for a while, but the increases in performance and the decreases in price over the last 10 years or so have really made it possible for it to be integrated into routine public health operations rather than just being specific research projects. In this blog, I've outlined what sequencing is, how some of the different sequencing platforms work, and also how we can utilize sequencing to help us monitor and control infectious diseases. There are, of course, many other uses for sequencing, but that would fill multiple books rather than one small blog. But I have added links to resources if you want to know more. I've also put a glossary at the end of the blog to help you understand the terms and the acronyms, and of course, scientists do love an acronym. First of all, what is sequencing? Sequencing is the method we use to determine the order of the four bases, A, T, G, and C, that make up DNA. Scientists were first able to read segments of DNA in the early 1970s, but back then, sequencing even small amounts of DNA was a very lengthy process. Nowadays, scientists are able to sequence vast quantities of DNA relatively quickly, and this outputs terabytes worth of data. This means that modern sequencing scientists need to have both laboratory and bioinformatics skills to go from a sample of, for instance, tissue, all the way through to understanding the final sequence. And you can read more about sequencing basics in the link on my blog. Now, a very short history of sequencing. The first real use for sequencing, or genomics, which is the study of genes, was DNA fingerprinting. DNA fingerprinting is still used to match unknown samples to a known sample, and it has been used by, for example, the police in crime cases as well as paternity testing laboratories. It was first used in a forensics case in the 1980s to identify the suspect of two rape murder cases, and you can read more about the history of DNA fingerprinting in the link on my blog. The Human Genome Project, which aimed to sequence the entire human genome for the first time, began in October 1990 and was completed in April 2003, and that single genome took 13 years. Prior to this, we didn't really know that much about our genome and what makes us us. And in the Human Genome Project, researchers aim to understand our genome in three major ways. Firstly, to determine the sequence of all of the bases in the human genome. Secondly, they wanted to map the locations of the genes on our chromosomes. And actually, a lot of our genome is made up of what's called junk DNA. Finally, they wanted to produce linkage maps so that inherited traits, such as for genetic diseases, could be tracked over generations and understood better. The finished human genome sequence is actually derived from the DNA of several different volunteers, so not just one person, and that single genome cost a staggering $2.7 billion. The researchers mainly used Sanger sequencing technology, which I'll introduce below, and this is why it took so long. Sanger sequencing was the original sequencing method, although many laboratories still use it, and it's very useful for answering certain questions. It's known as sequencing by synthesis because to identify the sequence of the bases, new DNA is added to the strand of DNA we want to understand. In very simple terms, the strand of DNA that we want the information about is divided into four separate reaction pots, all of which contain all four of the standard deoxynucleotides. So that's the A's, G's, T's, and C's, and these are just free-floating individual bases that are used to build up the DNA strands, as well as a DNA polymerase, which is an enzyme which attaches the free-floating DNA bases to the strand. To each of these four reaction tubes, a single terminating dideoxynucleotide, A, G, T, or C, is added. And the DNA we're interested in is split in half by heating it up, which is called denaturing. And when this special terminating dNTP is bound to the template DNA strand by the DNA polymerase, it stops any more bases being added. This is because it makes the DNA polymerase drop off a bit like the end of a zip. And what you're left with is four different pots, each of which has strands that end with an A, a T, a C, or a G, and all the DNA fragments are of sort of random lengths based on where the terminating base attached. The fragments are then run on an agarose gel. When an electrical charge is applied to the gel, the smallest DNA fragments move through the gel the furthest. which is because the gel has a honeycomb-like structure and the smaller pieces are better at squeezing through the holes. What you then get is a sort of ladder of DNA bands on the gel, and this shows the sequence of DNA. You might find it makes more sense when you actually watch the video on how Sanger sequencing works, and you can find the link on my blog. Next generation sequencing technology is the current sort of version of technology, and while Sanger sequencing does still have its uses, it can only sequence a single DNA fragment at a time. So you can imagine that sequencing the entire genome, such as in the Human Genome Project, might take some time. But as I say, technology has moved on, and the current era of sequencing is known as next generation sequencing technology, or NGS. Now, this is massively parallel, which means that scientists can sequence millions of fragments of DNA or RNA simultaneously per run, making it vastly faster than Sanger. There are a number of different next generation sequencing platforms, and there are pros and cons for using each of them. They can broadly be categorized into short read sequencing and long read sequencing. Short read sequencing, as the name suggests, utilizes very small fragments of DNA, usually hundreds of base pairs in length, and long read sequencing uses fragments that are thousands or tens or even hundreds of thousands of base pairs long. In fact, it's currently possible to sequence an entire bacterial genome of around 4 million bases in one single strand of DNA. Short read sequencing platforms include Illumina and IronTorrent, while some long read platforms include PacBio and Oxford Nanopore technologies. You can find information about how Nanopore and Illumina sequencing compare in the link. I'll explain a little bit more about how Illumina and Nanopore work, as they are two very commonly used platforms. Like Sanger, Illumina also utilizes sequencing by synthesis. In Illumina sequencing, DNA samples are cut up into chunks of 200 to 600 base pairs and then amplified by something called bridge PCR, so that there are lots of copies of those little DNA chunks. Now, these chunks are denatured. The two strands, as I say, are separated like unzipping a zip, and then new bases are added to that strand. But this time, the newly added bases are fluorescent. When each base is added to the template DNA strand, it fluoresces a different color. So, for instance, A might fluoresce red, and this is recorded by the sequencing machine. The machine can then work out, based on the sequences of the colors that fluoresce, what the actual sequence is. Oxford Nanopore Technologies, or ONT, or nanopore sequencing, is a platform that uses quite a different method to identify bases. A single strand of DNA or RNA is passed through a protein pore on a membrane. An electrical current is passed across the membrane, and every time a base passes through a pore, it changes the electrical signal. These resulting changes in the electrical signal are recorded and decoded by the computer to provide the specific DNA or RNA sequence. If you're interested in how the scientists came up with this technology, you can read a history of nanopore sequencing in the link. I actually thought, whilst it's quite a long article, it's really very interesting. Now we know a little bit about how it all works, let's go through some of the ways we can use sequencing for infectious diseases. Genomics is often seen as the more expensive option, but as technology advances, it's being rolled out in more countries and for more purposes. If you remember in the Human Genome Project, it cost nearly $3 billion and took more than 10 years to sequence a single human genome. Now, it's approximately $600 per genome, and we're talking hours or days rather than months or years to sequence it. For pathogens such as viruses and bacteria, which have much smaller genomes than humans, it's even cheaper and more rapid. A bacterial genome now costs as little as $40 and can take as little as a few hours to sequence. It's really exciting to think where we'll be in another 10 years' time. For diagnosing diseases, there are some advantages for sequencing when compared to other methods of diagnosis, such as culturing the pathogen or using molecular methods such as PCR. Sequencing can generally give us much more information about the organism that causes the infection, which in turn can help us to treat it more appropriately. Let's use tuberculosis as an example. And yes, I know I do use it a lot, but it's a very difficult pathogen to treat. The mycobacteria, so the family of pathogens that can cause TB disease, are generally slow to grow, and when you culture them, the different species that can cause human disease, also known as the non-tuberculous mycobacteria or NTMs, all look the same. Looking at the genome of these pathogens can reveal whether a person has a mixed infection, so different strains of the same organism, such as two different strains of M. tuberculosis, or more than one species, so say M. tuberculosis and M. avium, which might then be resistant to different drugs, making a person's infection harder to treat. Sequencing can identify these substrains and lineages of pathogens, and based on this, it can be used to identify if a patient has relapsed or become reinfected if they're not responding to treatment. For instance, if a patient with tuberculosis fails to respond to the often long and difficult drug treatment regime, we can use sequencing to identify whether that patient has relapsed. So the genome of their TB bacteria is the same at the beginning of their treatment as at the end, which means it's likely that it's the same bug and the treatment didn't actually kill it off in the first place, or whether they've been reinfected by a new, different M. tuberculosis strain, and this is especially common in high burden countries. Diagnosis using sequencing may include sequencing the whole genome or specific target genes of pathogens infecting individuals, or it may utilize metagenomics, where multiple genomes from a sample, such as human sputum, are sequenced, and this can be used to identify what might be causing or what pathogen might be causing, say, lung disease. Sequencing is also really useful for disease surveillance and for tracking outbreaks, because we can compare pathogen genomes isolated from different patients and work out how similar they are, and then we can trace how they move through a population. It can be effective at an individual level, such as an outbreak on a ward or in a hospital, or between patients in a community, or on an international level, such as during a pandemic. This was used to great effect during the West African Ebola epidemic and, of course, during the COVID-19 pandemic to track the SARS-CoV-2 virus. Surveillance, like diagnostics, may include extracting and sequencing the whole genomes or specific genes of pathogens infecting individuals, and then comparing them, or it may utilize metagenomics. Sequencing can also be used to identify whether a pathogen is resistant to the drugs we use to treat it, because a lot of drug resistance is caused by the presence or absence of a specific gene, mutations within genes, or a more complex combination. You can read more about how antimicrobial resistance, or AMR, works in the link on my blog. Sequencing can be used to identify new drug resistance mutations and novel drug targets. To conclude all of this, sequencing is a really important tool in the fight against infectious diseases, and as technology is ever improving, it means it's getting cheaper and more readily available. The integration of genomics into diagnostics and surveillance is really going to help us control and treat pathogens, hopefully reducing human and animal and plant infections and suffering. It's exciting to see what the future will look like.